Enterobacteria is a large family of gram-negative rod-shaped bacteria that are widely distributed in nature, especially in the intestinal tract of humans and animals. Identification of Enterobacteria is commonly carried out using biochemical tests, including oxidase tests, McConkey agar, indole test, and hydrogen sulfide production. These tests have differentiated between genera and species based on their metabolic properties. Therefore, this practice is important to understand the identification and significance of Enterobacteria in food safety and microbiological analysis. A positive oxidase test is indicated by a rapid color change within a second to dark blue or purple after the addition of oxidase reagent, while no color change indicates a negative result. For example, Pseudomonas aeruginosa gives positive result to oxidase test. Pseudomonas is a highly adaptable gram-negative bacteria. It has rod shape and known for its versatility and pathogenicity. It is widely distributed in natural environments such as soil, water, and plants, as well as human impact areas like hospital equipment and moist surface. McConkey agar is a selective and differential medium widely used for the isolation of gram-negative bacteria. The medium contains bile salt and crystal violet, which inhibit the growth of gram-positive bacteria, making it selective. This medium is commonly used in food microbiology to detect coliform and potential pathogens such as E. coli and Salmonella. The McConkey agar will produce red colonies and white colonies. The red colonies will continue the indole test to determine the E. coli. E. coli is a gram-negative bacteria. It has rod-shaped and facultatively anaerobic bacterium that is a natural inhabitant of the gut microbiota in human and animals. A positive result of indole test is indicated by the formation of red ring at the surface of the medium, while a negative result shows no color change or a yellow layer. The white colonies will continue the H2S production test used to determine whether bacteria can produce hydrogen sulfide gas through the reduction of sulfur-containing compound. A positive result is indicated by black coloration in the medium, while the absence of blackening indicates a negative result. The positive result will continue the indole test to determine the presence of Proteus vulgaris or Proteus mirabilis. Proteus vulgaris will give positive result to indole test, which is by formation of a red ring layer at the surface, while Proteus mirabilis will give a negative result by showing no color change.
The oxidase test begins with a briefing from the lab staff on how to perform the test. First, drop a drop of oxidase onto the paper. Then, took a single colony from the petri dish A using a toothpick. Spread the colony on the paper using a toothpick. Repeat the same procedure for another colony in petri dish B and C. To make streaks on McConkey agar, first lit the Bunsen burner. The colony used was the same colony from the oxidase test, so the colony was picked and streaked on agar. Colony from petri dish B was streaked on nutrient agar, while colony from petri dish C was streaked on McConkey agar. Then the agar incubated for 24 hours at 37 degrees Celsius. Took a single colony from the nutrient agar to step and spray into the H2S production medium. A single colony from the McConkey agar was taken and placed in an indole test tube. Then the agar incubated for 24 hours at 37 degrees Celsius. For the indole test, first heat the inoculation loop and cool it first. Next, took a single colony from the H2S production medium and put it into the indole test tube. Finally, dropped the Indole reagent into the test tube and wait until there was a change.
Today, I will be presenting the results for our experiment titled Determination of Enterobacteria C. Firstly, for the oxidized test, we obtained one positive result and two negative results. The negative results indicate the possible presence of Enterobacteria Ca bacteria because most Enterobacteria Ca are oxidized negative. Next, for the McConkey agar striking, we observed red colonies and white colonies. The red colonies indicate lactose fermenting bacteria, while the white colonies indicate non-lactose fermenting bacteria. Then, for the indole test from the red colonies, a red ring layer appeared after adding the reagent, which indicates a positive result. This confirms the production of E. coli. Next, for the H2SO4 gas production test from the white colonies, black colonies appeared after incubation. This indicates a positive result for H2SO4 production. Lastly, for the indole test from the H2SO4 producing colonies, a yellow ring layer appeared, which indicates a negative result. This confirms the presence of Proteus mirabilis.
In this experiment, Chica samples were used to determine the presence of Enterobacteria through several biochemical and selective media tests. The oxidase test show one positive result and two negative results. Enterobacteria are generally oxidized negative bacteria. Therefore, the negative oxidized result indicate the possible presence of Enterobacteria species in the Chica sample. The positive oxidase result may indicate the presence of non-Enterobacteria bacteria contaminating the sample. According to research by Farmer et al. 2007, oxidase testing is widely used to differentiate Enterobacteria from other gram-negative bacteria because Enterobacteria lack cytochrome oxidase enzyme activity. And then the McConkey agar streaking produce both red and white colonies. Red colonies on McConkey agar indicate lactose fermenting bacteria such as Escherichia coli, while white colonies represent non-lactose fermenters such as Proteus or Salmonella species. The presence of both colonies types suggests mixed bacterial contamination in the chicken sample. Chicken meat is highly susceptible to bacterial contamination during slaughtering, handling, transportation, and storage. According to research by Roger et al. 2017, raw poultry commonly contains Enterobacteria including E. coli, Proteus, and Salmonella due to fecal contamination and poor hygiene during processing. The indole test performed on red colonies produced a red ring layer, indicating a positive result for Escherichia coli production. E. coli is an indole-positive bacterium capable of degrading tryptophan to produce indole. The presence of E. coli in chicken meat indicates possible fecal contamination and poor hygiene handling. According to research by Ibrahim et al. 2020, E. coli is one of the most common bacterial contaminants found in raw poultry and serves as an important indicator of sanitation and food safety condition. Furthermore, the H2S gas production test from white colonies shows black colonies formation indicating a positive H2S production result. H2S producing bacteria are commonly associated with Proteus and Salmonella species. The subsequent indole tests from H2S producing colonies show a yellow ring light indicating a negative indole result and confirming the possible presence of Proteus microbials. According to research by Derzewika 2016, Proteus microbialis is commonly found in raw meat and poultry products due to the environmental and fecal contamination and may contribute to food spoilage and foodborne infection if food is improperly handled or undercooked. In conclusion, the chicken sample used in this experiment was contaminated with several Enterobacteria bacteria, including Escherichia coli and possible Proteus mirabilis. The oxidase McConkey agar, indole, and H2S production tests successfully differentiate the bacterial species present in the sample. The presence of these bacteria indicate possible fecal contamination and poor hygienic condition during handling or processing of the chicken. Therefore, proper cooking, safe handling, and good sanitation practice are essential to reduce the risk of foodborne illness associated with contaminated poultry products. That's all from us. Thank you.